We are live now.
Okay. Good evening, everybody, all the participants and distinguished guest. Today is the fourth day of our international webinar series, Unravelling the World of Nutrition, organized by the Department of Applied Nutrition and Dietetics, Sustainability University. And the whole session will be moderated by our faculty, Dr. Moumita Das. Over to you, Moumita.
Thank you, sir. Am I audible properly?
Yes, you are audible, Moumita.
Okay. Good evening, and I welcome you all to the fourth day of our international webinar series. Today we have Professor Oshim Dr. Roy Chaudhary with us. And before we could move on, I would like, we also have our Honorable Vice Chancellor, sir, with us. Professor Dr. Dhrubajyoti Chattopadhyay. So first of all, I would like to request him to say a few words before we move on.
Good evening.
Good evening, sir.
Good evening, everybody, and welcome, Oshim, again back to our webinar. And you know that this is in this nutrition month, we are having this fourth day of this international webinar. We're having distinguished guests from different parts of the world, and really we are very happy to have you again. This webinar is covering very interesting topics actually from in the field of nutrition. And I hope that this will be a great asset for the students of this nutrition and dietetics students. Let me first again thank Professor, Madam, professors and madam and students and participants, everybody, before we start our lecture. Dear students, the topic itself is very interesting today because this nutrition and gut microbiome, this gut microbiome is becoming one of the major area of research in the last couple of years with respect to now people working in the area of prebiotics, probiotics, and also regarding this gut microbiome biota is very interesting. And Oshim, you will be happy to know that one of the research work which is going on in our university by one of the tutor in nutrition department, no, it is actually one of the faculty Anindita Mukherjee from psychology department. She is actually trying to correlate with the different types of psychological problem related to the this gut microbiota. So that type of microbe, you know, that lactobacillus is one of the major ones, and the type and species of lactobacillus, which is actually going to give the persons also with respect to their level of different types of psychological problem. Now, in this respect, I can tell you that we have one of our faculty, Apri Ghosh, is actually looking at it from the Department of Microbiology. And today's lecture with respect to the in general will definitely help our research work also. So thank you, Oshim, for joining us. And I'm sure we are going to learn a lot of things from your lecture. Thank you.
Thank you.
Thank you, sir. Thank you for your words. And now I think Nilanjan, sir, I'll hand over the platform to Nilanjan, sir, Dr. Nilanjan Das. Sir, are you here? Okay, sorry for the interruption, sir. Actually, due to some technical glitch, Dr. Nilanjan Das is unable to join, I think. Today we have with us Professor Oshim Dr. Roy, and he is faculty of medicine from the University of Oslo. And he has worked on different fields, but today we will be hearing from him about gut and gut microbiota and its interaction with nutrition and food. So I think it's going to be a very interesting lecture. So over to you, sir, without wasting much time.
Yeah.
Yes.
Okay. Thank you, everybody. Thank you, especially, of course, Sister Nivedita University and its coordinator, Dr. Som Shubhra Thakur, for inviting me to share some of the latest development in this area. Actually, I must say in this context that in 2018 or 19, Dhruvada actually, the Honorable Vice Chancellor now, he actually organized, along with the Indian Microbial Society, a meeting in Calcutta. And he invited me to give a talk on this sort of aspect, microbiota and cardiovascular health. Actually, from that time, I started a little bit work in this area. And today I can say we have completed some of the biggest trial on this issue, which I'll be talking later on and something. So today, actually, I'm not a microbiologist, not an expert in this area, but what I will share with you, the different facets of this very interesting and what I should say, a very well emerged, recognized both in the medical field as well as nutrition field, in overall health and disease processes that we are in at the moment. So with this, I will share the screen and start my lecture. Just a minute, please. Where to start? Okay. Can you see the screen now?
Yes, sir. Yes, it's visible.
Okay. Here I am. It is a very sunny day, but temperature is very cool. In a Riks Hospital, that's a national hospital in Norway. And we work with several hospitals and things. As you know, probably Norway is as a social medical policy. That means everybody get free of charge any treatment, whatever it is, and including transport by helicopter or by car, whatever is there. So it's a 100% actually protected by the government here. So the government is making sure that everybody's health is very important, and they maintain the health. And to maintain the health means you have to understand your food, nutrition, environment, and physiology, so that you can really becoming less independent on drug or medicine, and have a very lengthy life, something like that. So with this, sorry, I have to, can I? Yeah. So since this talk is about today nutrition and microbiota, so I thought I would start with food. What is food? And as you know, food is a mixture of macronutrients, carbohydrate, fats, and protein, but it got a lot of essential nutrients, including vitamin, etc. And we take this food and process in our body, and that contain a huge amount of our own cells. Plus, we have 1 to 10 times bacteria, not bacteria, microbiome. It contains bacteria, virus, fungus, archaea, etc. That is also in our body. And also we have a lot of organs. They have a different demand for different food, and they have different machineries. They have, total we have 23,000 genes, etc. And we have but in our body, we have trillions of microorganisms. They got so many genes. So in number of genes, we are dwarfed by the bacteria that contain in our body. And also we get hundreds types of, 1,000 types of compounds of nutrient importance in nutrition, food, and also they have a biochemical, sometimes hormonal effect, sometimes effect that can change the epigenetics, etc., etc. and there are thousands of lipids, you name it. So all this is a kind of a niche that we are surviving ourselves along with our the bacteria that we host there. Sorry. So this is to just to introduce you that we have a human body containing 23,000 genes, but we have a normal flora, microbial cell that is trillions, and they got so many genes. So in terms of number of genes, etc., we are really very, very little contribution to our whole system. So this microbiota, that's why it is now thought that it can act as a metabolic organ. That I will try to introduce you to is a different kind of a research which is going on that involves our well-being, as well as it can, if it is gets a problem, then it can produce a disease, something, how they are cross-talking with our own system along with this microorganism. This is very important. So in a way, human, sorry, we are, what is going on? In a way, human body contains only 43% of our own cells, and less 57% are from actually some bacteria, etc. So I think there is some problem with, So when you see the visible organs, you can see the different organs. So different method we have, from MRI to all sorts of methods, and we know that many genes are there, but that is not complete ourselves. We have so many invisible cells. That is, it contains around 9 million of genes. This is spread all over the body. And that if we combine this together, then we are a complete human being and normal functioning body. I will show you a different experiment to come to this conclusion that for normal functioning body, you need to have a normal functioning flora, bacteria in different places. So our own genes among 23,000 genes, and microbial origins, it can range 2 to 3 million, but they constitute 1 to 3% of our body mass. And in an 80 kilograms of adult, the total mass of the microbes may fluctuate from 2 kilo to 3 kilos. So you can imagine, for this sort of microbes, we need to provide a niche that has their own nutrition, their survival, location, etc. It should be properly maintained. Oh, something happening. Actually, the gut microbiome story and the health, it is a long time back. Hippocrates says all disease begins in the gut. And then he said that health is determined by the microbiota in our gut. So I don't know, but it was, he said, it was very interesting that long time back, he understood that he had the gut feeling that the gut microbiota has a lot to do with the health, human health. Okay, I think something problem is there. So there are 50 bacterial phyla in the world, in the cosmos, whatever you call it, and 6 phyla in our body. And they are mostly Firmicutes, Bacteroidetes, and Actinobacteria, etc. And they are representing genera, as I mentioned these things. You can, but most important is Bifidobacterium and Lactobacillus. These are 2 very important that you have to remember while I'm talking. So it seems like that diet and food that you are exposed to ourselves every day, there are 2 parts. One is the digestion, uptake, and food components that is keeping our own cells happy and proper energy, etc., etc. But there are also non-digested food components. Also, there are something else, pollution, etc., that I'll come later on. That is actually utilized by intestinal microbiota, the gut bacteria, which is present, and it maintains certain diversity. And they actually work on these non-digested food components. And they produce so many metabolites that will come and that crosstalk with our human system with the bacteria. And that is actually very important. These metabolites produced by bacteria that can go to the blood and the circulation, and it can go to every tissue and can have a huge impact. So if the microbiota changes, then this, of course, its metabolite will change. Then we'll have a different kind of problem that I'll be talking later on. So we have a good and bad bacterial flora. Good bacteria, you can say commensal bacteria in terms of memorizing it. And the bad bacteria, it could be different Clostridium species, etc. And they produce certain pathogens and things, and we're having problems. And so there is a fine balance in the gut microbiota. One is the commensals. They inhibit the pathogen growth. They convert prodrug to active metabolites. It degrades polysaccharides of plant origin and produce short-chain fatty acids, etc., etc., and vitamin K, and stimulate and modulate immune function. It also very, very much control obesity. A person may nothing to do with the food intake, but the bacteria, if gone wrong, then the obesity can develop. And also is very important, the gut mortality is controlled by this. But there is other thing, that is pathogenic bacteria. They produce inflammation, liver damage, etc., and diarrhea, which I'll be talking later on. But this, how this microbiota actually ended up in our body, and it starts from the very early beginning. Actually, as we know that during utero development, that we develop ourselves as is a kind of a sterile environment. It is not true anymore. The bacteria can go through the placental barrier and can colonize the fetus. So there are a lot of studies showing that the early establishment of the bacteria in our body started in the mother's womb. And then, of course, it is the mode of the environment and the thing and how we are delivered, that is also important, the bacterial species that we get from the mother. If it is a normal birth canal delivery, then we get different sorts of bacteria from the mother. And if it is a cesarean delivery, then we get mostly skin bacteria from the mother. The babies get it. And also gestational age is important. Then the feeding actually determine the microbiota and the bacterial population, and is a family member interaction, maternal diet, etc. And throughout the life, there is a changes in the microbiota and the disease pattern and health pattern that I will be talking. So if you look at this slide, the microbiota, the population of the bacteria in the gut are actually subject to this sort of regulation. Firstly is a diet. If you take more refined food, that means they don't have enough food, carbohydrate, complex carbohydrate for the gut bacteria to survive on. And the environment pollution, they might get into them and they can change it. Antibiotic treatment from the early life to any time. It can also changes the microbiota in the gut. And also the way of rearing up, breastfeeding and formula feeding, they have also control on the microbiota situation. And prenatal and maternal factors are very important. The development and establishment of the gut microbiota. And also that I'll be talking about the antimicrobial peptide is produced by mucus is very important. It's also controlled by microbiota. All these things actually contributing to the microbial population that we are having as a young man, as a child, as an old man. So these are the factors. Actually, it is a very dynamic situation there. But also we now know, we have data showing that the gut microbiota and geographical location are involved. African children have greater proportion of bacteroides and gram-positive organisms in their bowels, whereas the Western lifestyles in the kids, those who are having those lifestyle increases firmicutes and gram-negative organisms. And now the research is going on to look to find out the relationship with the presence of different kind of microbiota in different geographical location. And another important thing is very important as a nutritionist, you must understand that not everybody is responding to similar kind of carbohydrate load to the glycemic response. And if you see this cartoon, it is the work done in Israel showing that the one population getting, see the above cartoon, that is banana and cookies, they have quite a different glycemic response with a similar amount of carbohydrate. They're given, but their glycemic response is quite different. And if you look at this population, bottom one is quite different. So that means there is something apart from the enzyme system we know that handles the carbohydrate is responsible for the glycemic response in the body. And also recently a study done with a different bread preparation, and it showed that depending on the bread preparation and bread carbohydrate, they have a different kind of glycemic response. So it is not true that if you eat a similar amount and type of bread, everybody will have a similar glycemic response. So there is a big difference of that. And we have 20 genes to metabolize any carbohydrate, whereas a bacteria has each bacteria in our gut, it contains 266 genes to metabolize the carbohydrate. And we have billions of them. So you can imagine the help that we get from the bacteria for the carbohydrate metabolism in the gut is tremendous. And also a lot of things we see, the changes in the glycemic response or personalized nutrition. Not everybody is responding similar way to the similar amount or similar type of food is because of the colonization of the bacteria, of different bacteria in different bodies. This is one of the factors. Even for personalized medicine, it is coming now like that. So, this gut microbe, actually, they have different functions. One of the function is they protect the gut. They actually fend off any pathogenic microbe. They don't allow it to grow there. It stimulates the immune systems. It protects the integrity of the gut. It synthesizes vitamins and short chain fatty acid. And of course, its major function is actually to degrade the complex carbohydrate and also proteins into smaller so that it can survive on that and it helps us. And also it modulates the effect of drugs. Some of the drug may not be, or medicine may not be effective because this bacteria is metabolizing different way. And you have to remember that these gut bacteria, they need at least 60 grams of complex carbohydrate every day for survival. And by taking different sort of food, you can really impact the gut microbial population. If you take more protein-based food, then these bacteria, those bacteria that metabolize the protein will be much more active than those who are very good in carbohydrate metabolism. So that means all these factors can actually change the microbial population in the gut. And now I'll be talking about this microbiota, their role in the health and disease processes. And we have a very symbiotic relationship with bacteria, this gut microbiota. I'm not talking about all other bacteria that is all over the body, but I'm just restricted now talk to the gut bacteria. And these gut bacteria, their first impact is the innate immunity and adaptive immunity. They are very much involved in this. More research is emerging, and I will present some of the cartoon on this. And if you see this, as I mentioned, this gut microbiota, we have 6 phyla, and they produce different kind of compounds. The gut microbiota, they are not actually attaching the intestine, epithelial cell as such. They are over the mucus, but they are staying there, and they produce different sorts of compound. For short chain fatty acid, acetic, butyric acid, propionic acid, and from amino acid, they are producing indole, and they can cross this epithelial, go to the bloodstream, and can affect so many cells from adipocyte to the brain. Everywhere they have an effect. And I will be talking later on this. And also they produce peptide, polysaccharide, lipid molecules, vitamins. GABA is very important. If you have a less of GABA, you get depressed. But this bacteria can produce GABA, and conjugated linoleic acid is very important for mitochondrial activation. And this compound produced by this microbiota from the left side can cross the epithelial cells and can go to the bloodstream, and they can affect all this system from immune cell, fibroblast, cells present in innate immune system as well as adaptive immune system. And also the gut bacteria can get this product also from our body. That is oxygen, nitrogen derived compounds, neurotransmitters, peptide, antimicrobial compound that we produce in our body. It can go and also can influence the gut microbiota. If you take, say, for example, some polyphenols or something, we have shown that you can change the microbial population in the gut. So there is a crisscross talk between the metabolites of the bacteria and metabolites of our body, and they meet in the bloodstream. And also this compound produced by us can cross the epithelial, can go to the bacteria, and can change the population. As say, for example, pollution, oxygen level, etc., can affect the microbiota. So in gut microbiota, the health system, it provides nutrient and also shelter. And it increases the metabolic activity of the host digest plant carbohydrate. I mentioned that complex carbohydrate, which you cannot digest, it goes to the large intestine, then it is processed by the gut microbiota, and it provides vitamin B2, B12, potassium, folic acid, and also it doesn't allow the colonization of pathogenic bacteria, etc. And you can imagine when you take antibiotic, the vitamin level production is low because this bacteria is no more because of the antibiotic torture, and then we feel weak and something. So you have to repopulate this microbiome again in the gut bacteria, in the gut situation. So if you look at this slide, that is the complex carbohydrate that is affecting this gut microbiota, Roseburia, lactose, Inspiria, etc. And they produce carbon dioxide, hydrogen, short chain fatty acid, and they can cross the gut and it can improve the inflammatory bowel disease and asthma and energy metabolism. And bifidobacterium is very important, and lactobacillus, these are the very two beneficial gut microbiota. They are involved in so many things, which I'll be showing some of the results later. It prevents even diarrhea, constipation, inflammatory bowel disease, and it also in skin disorder, it can help. And also some of the lactobacillus species can act on tryptophan metabolites and can act on aromatic AHR receptors and can increase the barrier integrity and improve the insulin sensitivity. And also I'll be talking about branch chain amino acid and trimethyl amino oxide that is produced by certain bacteria, which is detrimental to our health and can cause heart attack, cardiovascular problems, etc. And if we take lipid, large saturated fatty acid, omega-6, omega-3, they have a different effect on the gut bacteria, and that can involve in changing body weight and adipose tissue, fatty acid storage, etc. And polyphenols like present in green tea and red wine or grapes, that resveratrol, they can also affect the gut bacteria and also ultimately affect antioxidant, anti-inflammatory and cardioprotective effect in our body. So in innate immunity situation, there's gut microbiota, they are actually involved in pathogen-associated molecular pattern recognition. And it is very important for our innate immune system to work. It must recognize this and through their PRR system so that it can work properly. And it is shown that through short chain fatty acid that bind GPR and can affect and can train the dendritic cells to act on as a part of innate immune system so that it can determine which is pathogen, which is not pathogen, and which is, if it is pathogen, they try to degrade it and try to make it becoming antigen presenting cells, and then it can produce the antibody. So it's very important, this gut microbiota. It helps in promoting or protecting innate immunity. And this is from my book that we wrote that from different research that short chain produced fatty acid produced from the bacterial microbiota. It can cross the epithelium and it can regulate T regulatory cells is very important. that is a main switch in inflammation and immunity. It is also regulated by short chain fatty acid produced by the gut microbiota, and also it can affect also interleukin 10 production by macrophages. And also, as I mentioned, dendritic cells are quite influenced by short chain fatty acid produced by the gut microbiota, and that actually involved in TGF beta synthesis, and then Th17 cells are very important helper cell in immune cells. It seems like they are highly linked, associated with microbial production of short chain fatty acid. And also in the adaptive immune system, that is a specific immune system that involves antibody production. It seems like the commensal bacteria are highly involved in actually training these B cells, and to produce the plasma cells, it is highly involved, and it is the response to the vaccine or response to the immune challenges, how this the B cells become plasma cells. It is actually a lot of things are controlled by commensal bacteria. And commensal bacteria through short chain fatty acid, again, it can bind to the GP free fatty acid receptors, GPCR, and then it can stimulate FOXP3 and can involve, and also to TLR2, it can stimulate T regulatory cells. So you can see different immune cell involved in the immune function, B cells and T helper cell and dendritic cells, as well as T regulatory cells, are actually modulated by the presence of short chain fatty acid produced by this gut microbiota. So if I summarize this part of the talk, if you see here, the gut microbiota, non-digested polysaccharide that you get from fruits and vegetables, etc., then it will be metabolized by, it will go to the large intestine, and the gut microbiota will metabolize to short chain fatty acid. And you see this short chain fatty acid does so many things. This receptor GPR43, GPR41 or GPR109, depending on the short chain fatty acid, whether it's a butyric acid, propionic acid, or acetic acid. That's three acids are produced in our intestine, large intestine, by this bacteria. They have a different receptor system, and they will work through this receptor, and this receptor is present in immune cells as well as in adipose tissue. And through their binding, they changes the as I mentioned before, FOXP3 cell in T regulatory cells, they actually then produce interleukin 10 and TGF beta and do other things. At the same time, this short chain fatty acid actually can it can inhibit the HDAC, that is involved in epigenetic modulation of the histone deacylase enzyme actually, and they are affecting dendritic cell activity. And ultimately, this IgA production is very important, is actually the involving plasma cell transformation is also regulated by short chain fatty acid. And also very important thing is goblet cell. Goblet cells are very important in the large intestine. It produces the mucus and mucin, and it is protecting our gut. And it seems like the gut bacteria present in the in here, that is mostly by this commensal bacteria. This short chain fatty acid can bind to the goblet cell and then stimulate the mucus production. So if you have a less of gut microbiota, the good one, because you don't take much of polysaccharides, the refined food, you do take more refined food, not much of complex carbohydrate, then this goblet cell will not have much short chain fatty acid and cannot produce the mucus, and that is ultimately, it will produce a leaky gut and have a lot of intestinal disorders, including diarrhea. So it's very important that gut microbiota here, it plays that from it production of short chain fatty acid involved in immune function as well as the protection of the gut by producing the mucus secretion on the intestine surface. And another important thing is the transformation of CD4 plus bearing T cells is very important in our immune function. That is a native T for CD4 T cells. It has to be transformed into Th1, Th2, T regular cell, and Th17 cell. And it seems like this gut microbiota has a lot to do with this transformation. And as I mentioned, the Th1 cells are involved in cytokine productions, allergy, inflammation, whereas the Th2 cells are protecting against parasite, but they are through the different kind of cytokines, whereas the Th17 cell is control the infection, and T regulators is the master switch of the whole immune process response. And if we don't have this short chain fatty acid produced by this microbiota, or we have a disbalance of this short chain fatty acid, then we have a different regulatory situation, and that can affect the antibody production, whether responding to vaccine or natural immunity could be a big problem if we don't get the help of the commensal bacteria, which is also involved through their production, the different population of Th1, Th2, Th17, and T regulatory cells. So if we see this in this slide, the polysaccharide, the complex carbohydrate we get, it goes to the large intestine, and it is fermenting there by the intestinal microbiota, and then short chain are produced, short chain fatty acid, C3 to C4, acetic acid, propionic acid, butyric acid, and they actually involve in several functions, including epithelial integrity and immune function. Also, this polysaccharide can be degraded and can produce oligosaccharides, and that sometimes could be a problem of different type of polysaccharide that I'll talk about in the IBD case, actually, inflammatory bowel syndromes. So, but this sort of gut microbiota that we have it throughout the life, it can change the population because of different external factors, disease, diet, etc. External factors is, of course, a diet. Prebiotic and probiotic can help, whereas the antibiotic can have a detrimental effect. Illness, lifestyle, living environment, they can affect the density and diversity and activity of the gut microbiota. And if it is changed, then it will impact the health. If it is better, then health will be very good or maintained very well. And if there is a problem in the population changes and the diversity is less and less because of antibiotic and pollution and high fat diet, etc., that can lead to dysbiosis, that the normal flora is not there, and that produce the disease. And also the other factors are very important. Aging old people have a different kind of senescence happen in the gut, and the gut microbiota changes, of course, and also genetics. And stress is very important. It is shown in the maternal stress during pregnancy. Depending on the stress, the microbiota changes in the mother gut. It is also there, and they affect the children, the babies born from them. So all these factors, external influences, internal host properties, that actually changes the gut microbiota population. If it changes is too much, then it is dysbiosis, it leads to disease. And if the changes for good and much more diversity of the gut bacteria and proper number is maintained, then it gives good health. So dysbiosis and the disease, the gut, if there is a dysbiosis there, the malabsorption syndrome would be common. Malignancy, colorectal cancer is one of the reasons, because butyric acid that is produced from the complex carbohydrate by this bacteria is disturbed, and butyric acid is actually protecting, is killing the cancer cells through different processes, is very important. It is not there. Inflammatory bowel disease, gut become leaky because the protection is lost and the mucus formation is less, and it is a big problem. And diarrheal disease, and there is one important disease we know these days is called Clostridium difficile, the infection, and that is actually a pathogenic bacteria. They colonize the gut, and a huge problem could be happening. And this another form are non-mucosal disease, obesity and metabolic syndrome, malignancy, liver cancer, and autoimmune disorders, and chronic fatigue syndrome, periodontal disease, etc. And gut-brain axis, mental, that already Dhrupada mentioned that in the university somebody is working in this area, is another important situation can happen. So And it is shown that in the colon cancer patient, there is these two bacteria are present in high amount, Bacteroides vulgatus and Bacteroides stereochoris are there. And they, in the low risk patient, that is the Lactobacillus acidophilus and Eubacterium are present there. So there a lot of work at the moment is going on, and the research is evidence is not very conclusive, but the colonic flora seems to be a major environmental factor that modulates the risk of the colonic human beings. And there are a little bit mechanism coming here: high meat diet, that's a high amino acid, and that is metabolized by these bacteria, Clostridium, Bacteroides, and sulfur reducer, and they produce HS plus, and they can lead to carcinogenesis situation and DNA damage. And also this high amino acid can produce methanogens and also can have a cytotoxicity. And ultimately, these bacteria does not help much in the gut. They survive because of high protein diet, and they lead to infection of the gut and lead to the carcinogen. That is the mechanism. But this is the latest one I presented in my book that is published, that how the short chain fatty acid, mostly butyric acid, are actually involved in the protection of the colon cancer. And that is shown that if you have fibers and polysaccharides and they are fermented by the gut microbiota, they produce short chain fatty acid. And there are three types of short chain fatty acid, but butyric acid is very important in colon cancer, and it stimulates P21, and it that allows the cell cycle arrest at G1 phase and produce apoptosis. So there is inhibition of colon cancer. Another way is inhibits histone deacetylase and histone hyperacetylation, and that's why the gene expression is less. So that's why there is one mechanism how this gut microbiota actually protecting the colon cancer by producing short chain fatty acid. And a lot of research now come in terms of signaling how the short chain fatty is binding different receptors, actually producing this cellular signal which inhibits the carcinogenesis there. Another thing you know that inflammatory bowel disease is a very common now, and it is a kind of inflammation autoimmune disease condition that affect the colon and also small intestine. And it happens, of course, the weight loss, vomiting, diarrhea, mental depression, etc., can happen. And it shows that how this that colon protection is lost because of these gut microbiota problems. And as you know, that this IBD is mostly driven by T cells. As I mentioned, T regulator cell is very important. Is one of the master regulator of immune response, while the Th1, Th2, Th17 are involved in different cytokine productions, good for health, sometimes bad for health, but there should be balance between these two. And this balance is disturbed when there increased production of pro-inflammatory cytokine by T helper cells. And as I showed earlier, the T regulatory cells actually can be stimulated by gut bacteria by different short chain fatty acid. And so it is now known, it's called FODM diet, that contain more polysaccharides, a complex cell is called polysaccharide, and less refined food. They can actually stimulate T regulatory cells and can inhibit the pro-inflammatory cytokine production by these T helper cells. Another problem is obesity and metabolic syndrome that is very common, and it is a very interesting observation came that how this gut microbiota plays a role in energy harvest, storage, and expenditure. It has been shown in a study in Mexico, where in the highly polluted city, the energy intake is not much, but people become obese and metabolic disorder, metabolic syndrome, they develop ultimately cardiac problems, platelet hyperactivity, but in terms of the food intake and is not much. So then there is some doubt, what are the other factor that is actually making people obese? And it is shown that the gut microbiota, it is by itself, is involved in energy harvest, storage, expenditure. And it is shown also the very good effect of the gut microbiota in a situation where the mother is not getting proper nutrition or total good amount of nutrition, that they gut microbiota go for starving because they don't like to take the food, so the mother can get the maximum food possible during pregnancy. So the good effect is there, but it is also it is shown that the gut microbiota also play an important role in the body weight control. A lot of grants have been approved in European Union to study this phenomenon. The most important study came in germ-free mice. It is shown that germ-free mice are protected in obesity, but they develop different kind of disease. They have problems that I'll come later on why it is like that. And it is shown that the gut microbiota composition is quite different between the obese people versus the lean people. So there it is showing that there may be something is there with the gut microbiota that controls energy metabolism in us. This study published in 2006 in PNS showing that the different microbial population in the gut, obese mice versus lean mice. As you see, these two species, firmicutes and bacteroides, are different in lean mice compared to obese mice. So it's clearly showing that firmicutes are more populated in the obese mice than in the lean mice. So although we know human obesity is a complex situation, it is not easy to pinpoint one factors. There are several factors that are there. Of course, how we feel in terms of satiety and why it is created and why that some people develop fat around the tummy, others around the leg. There are a lot of research going on, but it is shown that gut microbiota has altered composition in the obese people, and their fermentation is a different pattern, and it has an increased energy harvest it has. And it is shown that short chain fatty acid are present very high in the liver, and brain satiety factor is regulated by this. And fatty acid oxidation is one of the problem. If it's less or more, could be problem. And they have a leaky gut. Also, the permeability is increased in the obese patient, and they have a PYY and GLP-1 less from the L cells. So why it is happening, how it is happening. Again, I took the slide from my book, and this is showing that, again, if you have a dietary polysaccharides that fermented by microbiota producing short-chain fatty acid, mainly three types of short-chain fatty acid produced: acetic acid, propionic acid, and butyric acid. And they have a different receptors. They are present in adipose tissue, they are present in immune cells, and also in the gut. And if they produce acetic acid, generally binds GPR41, there's a free fatty acid receptor, and they produce, they are regulating peptide YY, and they regulate the satiety. So you can imagine now how bacteria is controlling our satiety through short chain fatty acid binding GPR41. And if propionic acid is there and much more, they're binding GPR43, they're actually regulating a glucagon-like peptide 1 receptor, they're lowered. And that means they have it is a kind of energy building situation. And butyric acid is a receptor in adipose tissue, and they actually allow, they inhibit the lipolysis, they allow the fat accumulation, and they interfere the insulin signaling system. And the adipose tissue then store the fatty acid. And there is a fasting factors from adipose tissue, FIAP, and they are also regulating by microbiota, and they inhibit the LPL in the circulation, so that no free fatty acid is produced from the chylomicron dietary fat we take. It produces the chylomicron, and the chylomicron has to be delipidized. Fatty acid should be donated to adipose tissue, liver, etc., and then it become that's where we digest our fatty acid and triglyceride. But this LPL activity is inhibited by FIAP, and also some product from the polysaccharide from the gut microbiota. And what is the situation? If you take a functional food, we have a gut microbiota as a probiotic and also antibiotic. And if you look at here, this probiotic, or I should say the microbiota, they actually, if you have a fibers, indigestible polysaccharides, they produce short-chain fatty acid, and short-chain fatty acid, depending on the fatty acid, that can affect the thing I already mentioned in the previous slide, and they modulate gut leptin, peptide, and GLP-1. And through that, if it is affecting the satiety, that's a different situation, and it's affecting the insulin signaling. Fat accumulation will be decreased depending on the gut microbiota of what you have. Otherwise, it will be increased and then modulate the obesity. Another is antibiotic. They will destroy all these bacteria and then will take away all these good bacteria, and then insulin signaling will be reduced and inflammation will be more because of pathogenic bacteria will be colonized there, and they will raise their level plasma LPS, lipopolysaccharides, and immune cell will function, and you have a continuous low level of inflammation, and that is very bad for adipose tissue, etc. And there will be fatty acid accumulation, lipid accumulation, and this is a big problem. And if you the functional food in terms of polyphenol, etc., choline, taurine, etc., they can modulate the bile acid and bile salt metabolism, and depending on the expression of FXR or TGRF5, and that will determine how the glucose is metabolized and energy expenditure is there. So all these things are combining together will determine the body weight that you maintain regularly. Not only the food we take, but also the gut bacteria, and also the medicine, could be antibiotic or other things that are always modulating this. Of course, the functional food that we can change a little bit here and there and can change the outcome. No, sorry, this is already took. Another thing can happen if you have a lot of C2, C3, C4, that fatty acid can go to the liver and becoming palmitic acid and stearic acid or oleic acid, and they go to the circulation and can produce the energy, the fatty acid for adipose tissue and in the circulation. So you can imagine the undigested carbohydrate, diet and fiber, producing this from that by microbiota is producing short chain fatty acid, and this short chain fatty acid can also help to produce a larger long chain fatty acid, and then it become part of our lipid metabolism system. So this is a bit complicated, this slide, just to show you that how different factors is actually involved in the insulin sensitivity. And insulin sensitivity or insulin resistance, it can be lost at very early stage. There's the pre-diabetic conditions. It is not only physical activity, food, etc., that is involved, but also involved microbiota is highly involved in the insulin's regulation of the insulin sensitivity. As you see here, that undigested foods, carbohydrate, fermentation, short chain fatty acid can cross the epithelial and involve in insulin sensitivity. And some drug we take, metformin, that is for diabetic drug, they actually can stimulate the mucus degrading bacteria, and they can affect the host glycan sulfate and sulfate reducing bacteria, and can also increase the insulin sensitivity. This is one of the mechanisms. The metformin can also stimulate the insulin sensitivity. And during protein fermentation by bacteria, these are, or by itself, they produce different phenol and peracresals and branched chain amino acid. The red meat, the source, they actually reduce the insulin sensitivity. But sulfur amino acid may be protective against this. And a lot of vitamins, cobalamin, flavin, and folic acid, they are actually helping to maintain the insulin sensitivity. So I actually produced this slide to show you that the other factor, apart from lipids and carbohydrate, these factors also highly involved in maintaining the insulin sensitivity in us. Another thing important is gut microbiota and brain. As Professor Chatterjee mentioned, that the mental state is highly involved with how your gut feeling is there. Gut microbiota is highly involved in that, because they produce these short chain fatty acids and other metabolites that can directly impact the vagus sympathetic nervous system. It can also affect through the production of serotonin and etc. They can have a systemic communication with the brain function and so many other things. And if you look at the right side, here again, the short chain fatty acid can directly affecting the neural communication through vagal and sympathetic nervous system, affecting the behavior, and through gut peptide production that is maybe controlled by short chain fatty acid can affect the brain function. Also, some of the neurotransmitter, serotonin, short chain fatty acid can increase the serotonin production, and then you can feel much better that way. Although serotonin works differently, it is involved in gut motility, etc. Not exactly in the brain, but still, nevertheless, serotonin production 70% present in the gut, and a short chain fatty acid from the gut microbiota helping in that. Also, a lot of immunomodulatory metabolites are actually regulated by the gut microbiome, is actually changing the systemic communication and the brain function. And of course, the cytokines, tryptophan metabolism, they're affecting the hypothalamus pituitary adrenal axis, and through which the gut microbiota is always affecting the brain function through different kind of mechanism, living far, far behind the brain. But this is one of the reasons the gut, we call it gut, microbiome gut axis. Although gut is almost independent brain, we call it. It has independent nervous system, etc. It can do so many things by itself, control itself, but it seems like that it is also get a help from the gut microbiota in order to maintain the human, the mental status of the human being. And it is shown that this GABA is a gamma aminobutyric acid is very much involved in depression, etc. And serotonin, it is the gut microbiota is highly involved in their production, and thus that way it can involve in the communication of the brain with the whole body and the mental situation. Of course, the other thing is through pathogenic bacteria. LPS can impact the immune function, and immune function means production of cytokines, IgA. They are also affecting the brain. Inflammation of the brain, etc., could happen and can impact the brain function through the immunological axis. So it is very clear now that the gut microbiota, through their different products, neurometabolic products, or endocrine response or immune action, they can change, they can modulate the brain function. And if you look at this is how this mental health and behavior are regulated by diet and gut microbiota. It is like this, as I mentioned, it could be good diet or bad diet or high fat diet. It could be antibiotic, and it could be probiotics. That all are impacting gut microbiota. And that means they are impacting their metabolites, and they are impacting the immune systems. And immune system will produce cytokines, and the brain will be continuous inflammatory conditions and producing a lot of mental depression and disorder, etc. Because of the oxidative stress would be there. Another thing, they can crosstalk with the hypothalamic pituitary axis, and also the entering nervous system that, as mentioned, is very important nervous system of the gut. They also modulate this, and they also affect this thing. So, as you see, neuroanatomical pathway of the gut-brain axis that can affect the brain, as well as the intestinal mucosal barrier and blood-brain barrier to the products of the gut microbiota, whether affected by antibiotic, probiotic, or diet, can affect this microbiota, can affect the brain function. Another aspect is gut microbiota and the cardiovascular disease. This is very common now because of sudden cardiac death. And as I probably mentioned in different lectures, that there are risk factors, but non-risk factor is very much confirmatory, like high cholesterol level is a risk factor for cardiovascular. Is not true for everybody. If you go to the Mediterranean countries, where the cholesterol level is high, but they don't have much cardiovascular problem. And also, if you take the data from Framingham study that involved 5,000 people, that shows that the high the people, those who are having a cardiac attack and those who don't have cardiac, has a similar level of cholesterol level. So there may be something other factors that involved in this. And last 5-6 years, they developed there's new kind of product coming from gut microbiota that can affect whole cardiovascular system in terms of atherosclerosis development, in terms of hyperactivity of platelets, lipid disorder that is affected by trimethylamine. As you see here, trimethylamine is produced from red meat, phosphatidylcholine, and other amino acid containing phospholipids, mostly from choline. And this choline metabolized by the gut bacteria, and they produce trimethylamine. And then trimethylamine goes to the liver, and liver, they have a flavin monooxygenase, and they put the oxygen molecule here and produce TMAO, trimethylamine oxide. And these are the product. This is the product is very nasty. It involves in plaque formation. It involves in platelet hyperactivity. It involves in lipid disorders. And a lot of research going on. I'm also involved in some research in the prevention of TMA production, how we can inhibit the TMA production by changing the gut flora so that, not all bacteria producing TMA. So we have to look for which bacteria producing TMA. We have to stop it. We have to somehow inhibit it, or we can use, it is shown in FMO knockdown mice that they don't put TMAO because they cannot put oxygen molecule and their gut is fine. But if you have a more FMO, that TMA is then produce TMAO and then have a cardiovascular problem. So it is very important that we can control this TMAO production either at the level of the gut or at the level of the diet or in the liver. Somehow we can find the inhibitor that can stop the TMO production so that our body is not exposed to TMO, the platelet, endothelium, surface, lipids, so that we don't develop atherosclerosis and lead to cardiovascular disease. And so the thing is that if you have a red meat and things like that, that contain dietary choline, betaine, and carnitine, that can, through the gut bacteria, they metabolize to trimethylamine, and then it goes to the liver. It produces FMO through the FMO3, flavin monooxygenase 3. It produces a TMAO, trimethylamine oxide. And this product goes to the blood circulation, and it is involved in insulin resistance. It is involving diabetes. It produces lipid disorders. It is a kind of a express CD36 scavenger receptor. So monocyte macrophage then take all the oxidized LDL and produce foam cells, and atherosclerosis processes start. And we are working on how it is affecting platelet hyperactivity. Platelet hyperactivity is very important, and because everybody can have an active, highly active platelets, and hyperactive platelet means produce certain compounds that activate endothelium and produce atherosclerotic processes. But you cannot make it inactive by taking aspirin, because aspirin cannot be used regularly because it has a lot of side effects, and 30% people are not active. So we have to find out some mechanism to make sure the platelet are tamed. It is not hyperactive, so it does not take part in the atherosclerotic processes, because if it does, then there is a problem. And also it is involved in high blood pressure situation, and also it is involved in obesity. So you can see if you take blood pressure medicine, beta blockers, it doesn't work because some people will be resistant because certain portion of TMAO will be produced and can change the vascular tone and can produce the high blood pressure. So if I summarize this talk, that protein-rich diet and these things that produce TMA, and TMA goes to the liver and produce TMAO, and then it is affecting everything from endothelium cells to platelet and systemic circulatory platelet, etc., and platelet hyperactivity. And also it increases the forward cholesterol transport, and it inhibits the HDL-mediated cholesterol transport, reverse cholesterol transport. It doesn't allow, and that's why we have atherosclerotic plaque formation, and it involves in hyperlipidemia, adipose tissue inflammation, and brain inflammation, and intestinal permeability, etc. And ultimately, a cardiac system failure, and it can lead to brain disorders and atherosclerotic and also thrombotic disorder. So there are different mechanisms involved. I don't have much time to discuss it, but to show you that if TMO is highly in the blood circulation, they work in different cell, different way. Atherosclerosis situation, they increase the vascular become one, and atherosclerotic process is increased. Platelet hyperactivity, I already mentioned, is involved. And through MAP kinase and NF-kappa B, they increase the pro-inflammatory cytokine production, and it is also reduce the anti-inflammatory cytokines. So it is bad actually. It increases the pro-inflammatory, decrease the anti-inflammatory situation, and under immune part, under immune part, as I mentioned, that increases CD36, so it allows oxidized LDL uptake, produce foam cells, and atherosclerosis situation is formed. And also in monocyte macrophages, all these things are active, highly increased, and a big problem for atherosclerotic situation. And inflammasome is increased, so caspase-1 and procaspase-1 is increased, and cholesterol metabolism is disturbed. So if you take care of all these global effect of TMO, it is involved in almost all the important cells involved in inflammation and immunity. And reverse cholesterol transport is also inhibited by TMA1. That means you have more cholesterol in the circulation, atherosclerotic condition will be prevailed. But there are a lot of research going on, and there are some drug is developed, and we now know some of the dietary situation, how we can help. Of course, the idea is to decrease the TMO in the body. And as you see here, high-fat diet is always increased TMO, is bad for the thing. Low-fat diet and Mediterranean diet is helping us to reduce the TMO. And it is sphingomyelin is very good to reduce the TMO, whereas the L-carnitine is not good. And lifestyle intervention, if it is needed, hypocaloric diet and exercise is important. And then, of course, as I mentioned, the gut microbiota is produced TMA, and but TMA lyase is important. And that TMA produced TMAO in the liver, and TMO actually then does so many things. And this flavin monooxygenase that is present in the liver that produce TMAO from TMA, there are 2 drugs are now available that can be used to reduce the TMA level, and also TMA lyase is inhibitors are available, and that the TMA cannot go to the FMO through if lyase is inhibited, and PSC and other things are there. And some of the antibiotics it is specifically working on the bacteria that are producing TMA can be helpful. And in this case, our recent study showed that some of the polyphenols actually increases the population of the bacteria that can produce less of TMA. And that is very helpful. And it is present also in red wine. In that way, one can decrease the production of TMA, and so you can decrease the production of TMAO. So resveratrol and berberine present in blueberry and resveratrol in grapes, and that can actually increase the bacteria population, which actually produce less of TMO. And this is very helpful in order to reduce the cardiovascular disease. So another thing is that the gut bacteria is involved in blood pressure regulation through different mechanisms, serotonin, dopamine, and norepinephrine production. TMO is a bad thing, and indoxyl sulfate and paracresol sulfate coming from proteins is also increase the blood pressure. Sodium, potassium metabolism is decreased by gut microbiota is very good for the maintaining the blood pressure. Inflammation is another thing involved in blood pressure. Gut microbiota can help in that. Of course, the short chain fatty acid is very important in regulating the blood pressure. They are involving 3 different receptors, GPR41, 42, and this is the latest established last year. Another receptor is important. Through short chain fatty acid, we can regulate our blood pressure. And this is the mechanism. Mechanism is that short chain fatty acid, mostly propionate and butyrate, they bind GPR41, and there is a vasodilatory reaction, and blood pressure will be lowered. And if an acetic acid, it can bind alpha 78, and that lead to vasoconstriction and renin production, and that can lead to blood pressure. So if you remove this one, then there could be vasodilation can present. Another thing, if there is a gut dysbiosis, the different bacteria are populated because of antibiotic pollution and fatty acid, saturated fatty acid, and that lead to inflammation. L-arginine will be less, and less nitric oxide will be produced, and less nitric oxide means high blood pressure. Similar thing happened in endothelium 1 production because of more of oxidized LDL, and that led to vasoconstriction. So you can see these are the modus operandi, how this gut microbiota controlling our blood pressure. And if you take a blood pressure medicine, it will not be that helpful because of this system will still work and can change the blood pressure pattern. And that's why some people are resistant to blood pressure drug, because we are now having a more knowledge how the gut microbiota and the diet is helping us to maintain the vasodilation or vasoconstriction. And as I mentioned, that butyric acid itself, the produced from high fiber diet, can work through G protein coupled receptors, histone acetylation, mitochondrial activity, modulation of microbiota. And there are several diseases because of lack of butyric acid, metabolic disorder, insulin resistance, Parkinson's disease can be linked with gut microbiota butyric acid production. And it is shown that butyric acid involved in histone acetylation regulation, and those are involved in this sort of brain disorder as we know today, Alzheimer, Parkinson, Huntington disease, and stroke. And mitochondrial activity is very important for the brain energy things, and it seems like that butyric acid actually provide these sort of things, but the butyric acid is less, then there could be a problem of this. So the fiber is very important for those sort of patients. And a lot of research coming on autism and psychological disorder, mental disorder. In terms of microbiota, we can target which microbiota is helping, and we can increase the population by stool microbial implantation. And there are several mechanisms and metabolites that is coming from microbial metabolism. Polyphenols, vitamins, short chain fatty acid, polyamines, they're all involved in epigenetic modification, and they can be involved in repression of pro-inflammatory cytokines, prevention of obesity, healthy aging, depression, and anti-cancer gene, etc. These are involved. So we are in this box now, and we are trying to find out which population and which species actually helping us in different way, and then we can selectively can we implant and transplant and can do this sort of things. These are the main research is going on at the moment by manipulating gut microbiota, both probiotic, prebiotic, and transplantation. So we can really, through this mechanism, we know that they do changes this sort of disease disorder. So I'll end this lecture with this slide, that we should be making sure that our co-traveler, that is, we have that number is much more than ours, is very important, that you get from the mother and throughout the life activity. They need a food, and they mostly of course protein they also like, but important is a complex carbohydrate, and that is that we have to make sure that 60 grams we need every day to maintain these 2 kilograms of microbiota in the gut. And this is very important, that they are happy and they produce right kind of molecules, and we maintain the right kind of species or the diversity, so that then we have important microbiota symbiosis, and that help us in terms of healthy aging, homeostasis, and maintaining the health. And if we don't do this thing, if we don't take whole grain cereals, vegetable, fruits, if we go for refined grains, high fat diet, and we take a lot of antibiotic, then we have a problem from systemic problems to brain disorder and mental problems. So it is very important that, which I try to highlight, to go into different aspects of gut microbiota, how important it is. And last 10 years, with many thanks to Barack Obama, he actually granted millions of dollars to study these unseen friends, which are neglected a long time back. Although from previous, the Greek big man, we know that this is very important gut to maintain the gut microflora by taking complex food is very important. Now we know how it is doing it, the mechanism behind it, and how we can maintain throughout the life. And also one thing I did not mention, that is gut microbiota population, the changes through the life cycle. So we have to make sure that we get the good proper food and optimum food for those gut microbiota, no matter what we do in terms of taking food and maintain ourselves, taking the food that mostly we recommend Mediterranean food that contains a lot of fruits, vegetables, etc., antioxidants, and olive oil that contain also other polyphenols. That all these things are very important if you want to keep yourself body fit, mentally healthy, happy, and for healthy aging. Thank you very much.
Thank you, sir. It was really an enriching session. And I hope all our participants will definitely have something to take back home. I'm sure they have learned something. But there are a lot of questions, sir. I hope, can we take up some questions?
Go to the screen now. Yeah. What time now? Oh, my 4 o'clock. I didn't see the time. Sorry. Hello.
Hello. Yes. Am I audible? Yes.
Yeah.
So can we take up a few questions?
Yeah. And if there are more questions, all are welcome to send through email or through you WhatsApp, whatever.
Okay. Thank you, sir. I just wanted to say one thing, that there are so much of interactions between the host and the gut microbes. The real question is, who is the host and who is the parasite? Like, I really feel so. So the first question that we have, sir, is from one of our students, Urbi Roy. She's saying, okay, first question is, what is antimicrobial peptides?
Okay, our gut itself, they produce antimicrobial peptides, and they actually kill the pathogenic bacteria, etc. So and stop the proliferation and growth of the pathogenic bacteria. So that is produced in the gut, and it's very important. They should be there. Okay. And there is a mucin and mucus they produce in the gut, so that it can make an envelope, so that the pathogenic bacteria cannot go into there.
Next question is also from her, which is very interesting. Does mutation occur in microbiomes? If it occurs, can new variants be formed or new variants be converted?
Yes, of course, we expect mutation. Any organism, they go for proliferation, and depending on the rate of proliferation, mutation is there. But we are much more interested in the diversity of the population than mutation itself, because it is so much, and you need a high technology to go through this. So at the moment, we are not interested in the mutation. Mutation should happen and would occur. But the important thing is we are looking at the changes of the population, whether we have a more commensal or less of commensal, and the diversity that we expect. Even the latest study showing that the people who live very much long, we want to see their gut population and try to, transplant or to the people who are, under risk. So I think the research at the moment is to maintain the flora that we believe are healthy. But of course, mutation will be there, but we don't know much about it. Yeah.
Okay, so next question is, what is the relation between prebiotics, probiotics, and postbiotics?
This is a it's not exactly a scientific term. It is used by business people and also those who are making things up. Probiotic means all these bacteria in the yogurt, etc., that we're adding it. And a lot of people didn't believe that they could survive in the stomach and go to colonize the gut. But there are certain evidence it may be, but there are a lot of controversy there. But I did not show you, there are a lot of meta-analysis showing the effect of using probiotic. And prebiotic means their food, like polyphenols, beta glucan, etc. They actually is not absorbed in the intestine. It bypasses and it goes to the large intestine where it is being used by the microbiota and they proliferate. And we like to target those microbiota or species which are beneficial to our body. And that's why a lot of research and funding going on to find out both prebiotic, which there are a lot of species now. Lactobacillus is the main species, but there are a lot of species through mutation and through, I should not say mutation, through development, you can say. We are involved in this, which I say, for example, we found that some of these bacteria that can degrade the TMO in such a way it cannot go to the liver and can produce TMAO, or certain bacteria which will not produce TMA at all, doesn't matter how much red meat you eat. So it is possible. And other thing, I forgot the other question was, what was the other aspect of the question?
Prebiotics, probiotics, and postbiotics.
And these people give a combined both pre and probiotic, and it is being sold. And postbiotic means it is not much used in Europe, so it may be somebody's term. It's not very popular. Postbiotic means to maintain the flora that you have it, and if you believe that you attain this thing, and after taking antibiotic, etc., one can. It is very tough research to examine the flora that it is normalized or not, and then try to keep that. Maintaining this, it could be there, and it could be almost same like prebiotic kind of things, but we call it postbiotic.
Next question is, after consuming antibiotics, can our body generate the same species and same number of gut microbes which we possess at the time of birth?
I think at the time of birth, what we get mostly from the mother, and this is mostly 5 species, and then we get it from from the environment and also from breast milk, etc., etc. So they're changing, but the species remain the same. But antibiotic, depending on how much torture you are going through, and what is the changes happening, and there is of course there will be a situation when the antibiotic will be taken off. If there is number is there, but less number, they will try to increase. But one thing I did not say before, that is very important. I think it is not in the remit of this talk. That is called immunity genes. These bacteria that is surviving in the gut, they actually share the genes among themselves. So they know which is pathogen, which is not pathogen, and they try cross-talking with our immune systems. So through innate immunity, TLR system or PRR or other system, so pathogenic bacteria will not grow that much because of this system. Our innate system will kill them. At the same time, the commensal will survive and proliferate, and that kind of things that you developed with the fellow traveler. So that is a thing, but it's a complicated thing. Still, we are understanding how these bacteria survive in our system, those who don't have pathogenicity. It is very important. And it seems like we like them. And there is some kind of symbiosis, or I should not symbiosis, it is a commensal that is going on. But crosstalk is there through innate immune system. So there is a big chance of getting back to that, the bacteria that before the antibiotic was introduced.
So the next question is, does gut microbiota count changes with age?
Yes, it is. It is. I did not show the slide because there is no time there. It does. Throughout the life cycles, it changes. The diversity changes, population changes. It does actually from the time we are born to until we are dying. There's always changing going on, and there is a senescence could happen, and a lot of immune problems and things like that because of changes in the gut microbiota.
Okay, so the next question is, what is the role of firmicutes?
What is that? I didn't see that.
What is the role of firmicutes? Is it good or bad?
Okay, it is mostly bad actually, because they produce so many nasty things. Yes. Am I?
Yes.
I couldn't read anything. Is it? Yeah.
Okay.
Yeah.
The next question is that does the food additives and preservatives affect the gut microbiota?
Everything will affect gut microbiota. Depends how they are and how there is enough research to say that. But I believe the environment we live in and anything we inhale and take, if it goes to the gut, it will have impact, like antibiotic or anything. So it will affect, but whether bad or good or will increase or stimulate and what product they will produce, it is not much research yet. But there is a chance that it will affect, of course.
The next question is, how does gut microbial niche influence the steroid receptors, both plant and animal steroids in human body?
Okay, and that is AHR, actually, that is from protein, tryptophan. They produce indolic acid, some compound, and they can modulate the AHR receptors. And the AHR is involved in aromatic metabolism, etc. So that way it can affect. In the plant, I don't know, but in human and this cell culture system, it is shown it can.
The next question, could the baby's gut microbial composition be affected if the mother keeps changing her place of living during gestational period?
Yes, it is possible, and it happens. And because we now know that we are not always not in the sterile environment anymore, the bacteria can pass through the placenta and can go to the fetus. And if the mother changing the geographical location, their food is different, and there is enough opportunity for the bacteria to go into the system, it is possible, yes.
Okay. How the gut microbiome related with inborn disorders or syndromes?
Inborn syndrome is mostly genetic disorder, so I don't think there is much chance for gut microbiota to say anything. But of course, in the adult life, it is shown that in some time, especially skin disorder, rather, gut microbiota might help. But inborn disorder is completely genetic disorder.
Okay, the next question, how anti-nutrient factor increase or decrease gut microbiota?
Question is what you call anti-nutrient. One is called phytic acid, phytate, that is present in wheat and others. And phytate, it can go to the large intestine and can affect the microbial population. But not much study is done yet. But obviously, it is a matter of concern. But anti-nutrient in terms of how it works with binding different essential nutrients, but when it comes to the gut, it doesn't care. It has a metabolic, it tries to, like medicine, it also metabolize. Medicine, sometimes medicine become inactive, sometimes it can produce some derived drug, which can be helpful, can be bad also. So we don't know, but there is a chance of bacteria to metabolize these things, unless there is a xenobiotics going on.
Okay, so the next question is, what are the immunological tests we should perform to check the efficacy of a probiotic against growth of pathogens?
Okay, I think very simple test. If it really does it thing, you have to look at the cytokine profile in the plasma, blood, And also you can see the immune cells, whether the population changes or not. That is the best way to do. Then look for gut microbiota population in the stool and things like that. I think we should look for, even CRP could help this, CRP level, C-reactive proteins. We can have a 24-21 go, measure all these cytokines in anti-inflammatory and pro-inflammatory, and you can profile this, no problem.
The next question is, will intermittent fasting affect the gut microbiota?
Sorry?
Intermittent fasting.
Okay, a good question and very interesting question. We know very well that the microbiota's energy relationship is very interesting. If we take, it is very much involved in the weight regulation, body weight regulation, and it depends on which microbiota is involved, and it depends on the polyphenols and other things. And a lot of I was involved in the committee, approved some of the grants to look into the non-caloric nutrients that is involved in weight gain or weight regulation through microbiota. So it is possible that like a Swedish system, 5x2, that 5-day food, normal 2 days less food, that can affect. And also you can have a longer things, but for that, not much statistically significant research is done to conclude anything, but definitely it has some impact on the gut microbiota population and activity and colonization, the fasting. Whether it's for short-term fasting or other, we don't know, but it is there, especially in relation to adipose tissue secretion of ghrelin and leptin and other factors are involved in crosstalk between this, as well as a new factor is coming, FABP4 is highly involved in this sort of things.
Is there any action of hedonic food intake on gut microbes population?
I don't know, because there again, the last 1 and 2 years, it is a very unfortunate, not much research going on, but I have no idea. I cannot recollect it.
Okay. A similar type of question is asked about keto diet. Can keto diet affect our gut health?
Of course, keto diet means more fat and less carbohydrate, and you have more acid to acid kind of things in the blood. And it is important in epilepsy and other situations. And it might affect the gut population, bacterial population, but I don't have the data at the moment, but definitely it is things to pursue. How this, especially the effect of gut ketogenic diet in obesity, as well as in the epileptic condition, how the microbiota is actually doing under these two conditions. It will be important research point. Yeah.
Okay. Do the microbes work in the same way like genes, like we have genetic issue?
You see, we express 1% genes or less or something like that. And microbial genes are huge. We are dwarf in terms of number of genes that we have 23,000 and they have millions of billions. So really this issue depends on the study we call omics, that what the metabolites to differentiate between the microbial population versus our own. And also the drug and medicine we take it's a humongous task. It's not easy, but I think in future it will come when we have a proper handling of the cell signaling, the in vivo system, imaging we don't know, but we are going in that direction, of course, but it is a huge problem at the moment technologically.
The next question is, sir, is there any other medications that can change the composition and function of gut microbiome apart from antibiotics?
Any medicine will do that. Metformin will do that. Polyphenols does that. there is always some kind of balance will go on. They will try to even our blood, the products in the blood coming from our own metabolism can affect also microbiota. So there is a chance always, but that may not translate into real always the effect you want to see, the phenotypic situation. But obviously, there is a crosstalk going on between the gut microbiota, its metabolite, versus our own metabolites and our cells. So it is going on all the time. It is there. Whether there is a crosstalk between the brain, etc. Like in the plant, they produce millions of volatile compounds. In the daytime is different, nighttime is different. And plant crosstalk, when the plant is infected by insect, they send different kind of signal, volatile compounds, and it goes to the other plant, and then they crosstalk each other. And I think the volatile compounds in our body, it is coming a major issue now in terms of food and nutrition and our own upkeeping. The volatile compounds in the blood is very important, and it is getting more attention now. Yes.
Okay, so the next question we have is from Professor Shonjit De. He has asked, I would wonder whether you would mention any specific clinical biomarker which might act as an indicator for gut health.
Okay, very good question, Shonjit. I think, the gut health, we still use some of the marker, but it is very difficult at the moment to find out appropriate marker for gut health. We generally see the symptom and then we treat it. And of course, certain product, I believe the volatile compounds is coming that can be used as a marker, not a clinical marker, but early biomarker, early marker. It is possible. But at the moment, like IBD or other bowel syndrome disease, etc., we go through the mostly immunological marker because immune cells are highly important in that cases. So at the moment, it's difficult to say which marker should we use for the gut disease, except the known chronic diseases we know of, or like IBD and LBD, etc., and also bile acid could be used, but again, it is not that good marker yet. Sorry for that. We can't say much on this.
So the next question is, will the COVID-19 vaccination have any effect on gut microbiota?
Of course, COVID-19 vaccine or infection by COVID is the same phenomenon. That is, will be taken up by innate immune cells, and they will digest it, and then give it to the MHC1 to activate the cytotoxic T cells, and it gives to MHC2 to produce the antibody. And for that things to enhance gut microbiota would help in stimulating the innate immune system, as well as it will stimulate the T helper cells so that it can induce more B cells to produce becoming plasma cells. So it may have a direct impact on the production of antibody and the production of cytotoxic T cells and other T cells. It can help, of course. But as such, changing the flora, I don't think so. It is like that. But it will be, in terms of producing antibody and training the immune system, this microbiota would help, definitely.
Okay, so the next question we have is from our HOD, sir. TMAO reductase can change TMAO to TMA. Can any other gut microbiota produce this enzyme?
TMA ligase is there, but I think at the moment I'm not so sure about it, because the concern is mostly on FMO and TMA producing or less producing of bacteria. That is, we are more interested. And of course, it's possible, genetically modified, but then again, it will be GM. So it is not possible in that sense. But I don't know. It could be there, but no idea.
can gut microbiota increase the function of gene related to autism spectrum disorder?
That is a story not sure yet, and I don't think it will be done because it has a different kind of mechanism, autistic disorders and gut microbiota. There are some study done showed that transplantation of this can help and through different mechanism, but not this way.
So I think that's all. Do we have any other questions, sir? Shamshuddin, sir?
Dear Momita, I think we have covered all the questions, sir.
Okay.
It is
Okay.
always a great pleasure to be there in your talk, in your sessions. Today was the experience was also same. Thank you, sir, again.
Thank
I
you.
hope uh, we have talked earlier that whenever you will come to Calcutta, I hope you will
Sure.
visit to our campus. And this time I want to repeat it again. And now
Sure.
I want to ask our HOD of microbiology and biotechnology, Dr. Rajat Pal, to deliver the vote of thanks.
Hello, am I audible?
Yes, you are audible, sir.
Yes. Okay. Good. So this is a very big opportunity for me to give a vote of thanks to such an eminent personality. And first of all, I am thankful to Dr. Thakur Chaudhary to give me this opportunity. So first, I just want to thank Professor Oshim Dottoray for giving such a, wonderful lecture. I was just listening to it, and it was seriously mesmerizing. I have learned a lot, though I have done my all the studies in microbiology, but still today I was feeling that still I am a student in front of such an eminent scientist. So, Professor Dottoray, it's a very, very big pleasure for you to have with us. And as Dr. Thakur Chaudhary told, I will also repeat that whenever you will come to Calcutta, please do visit our campus. It's a very beautiful campus and very nice. So kindly have a look on it. And secondly, I will thank our Honorable Vice Chancellor, Sir Professor Dhrubajyoti Chattopadhyay, for giving us always the inspiration and enthusiasm to organize this kind of webinars and to introduce and interact with the other people. So I'm very much thankful to Professor Dhrubajyoti Chattopadhyay also. I'm thankful to Dr. Madam Ina Bose and her team to organize this webinar online in this online platform. I'm thankful to Dr. Thakur Chaudhary for heading the department and to organize this webinar series for so long time. And all the other faculty members, Nilanjan sir, Momita and Beepsha, everybody, we are really very much thankful for being with us, and all the participants, my dear students. Uh, be with us and enjoy the sessions and interact with the eminent professors coming around the globe. So that's all. I hope that for today, this is enough, and we will see again tomorrow.
Thank you.
Yes.
Bye-bye.
Yes, thank you. Thank you, sir.